you need to know. So first of all is on the structure of this protein. You have these primary, secondary, tertiary, and quaternary structures of proteins and relate to the protein functions and differentiate between hemoglobin and myoglobin. And then describe the classification of protein and amino acid, explain the chemical and physical properties of protein, discuss the deuterion and isoelectric pH of their importance, and then explain the glycoprotein structure and their functions. So basically, if you still remember, yesterday I talked about glycosaminoglycan and glycoprotein. So it's the same. It's just a bit continuation from yesterday on the glycoprotein. Okay, this definition, proteins are high molecular weight peptide made of amino acids linked together by the peptide bond. So protein is a big molecule, so the main small molecule or the build-up is from the amino acids linked together with the peptide bond. Okay, the importance of the protein, okay, as you know that the cytoskeleton is the internal protein network. Those are the cellular give the cellular shape and the integrity. And then your muscle also made up from protein, which is actin and myosin, is for contraction. Okay, hemoglobins for transport of oxygen. Same with the myoglobin, but myoglobin mainly is to store of the oxygen rather than transport. Then your antibodies, immunoglobulins, and complements, all almost all of your immune system enzymes also are all made up of protein. And then enzymes, most enzymes are made of protein. Okay. Then your receptors, some are glycoprotein, glycosaminoglycan, some are mainly protein. And then, of course, your molecular fingerprint. So your DNA, and then your serum albumin, okay, to maintain the fluid balance. And then prothrombin and fibrinogen for blood clotting. They also have the cytochrome components of your respiratory chain. One of the component in the electron transport chain, okay, cytochrome. And then protein also can be catabolized and give energy. But the energy that it gives is not as same as the, like I mentioned yesterday on the fats. Fats give up most of the energy, but protein is only not as much as fats. Okay, the structure. So proteins are made up of carbon, hydrogen, oxygen, and nitrogen. Is a polymer made of monomers, which is amino acids. Okay. So this is the structure. It's made up of carboxylic group, and then amino group, then hydrogen. Okay, and then this one is there are 20 different of our group, different group, which differentiate between the amino acids. So in total, we have 20 different types of amino acid. So this is how it forms by the condensation reaction. Okay, so this polypeptide is a chain of amino acid. So peptide bonds between those amino acid. So peptide bonds is considered the backbone of a structural framework for the protein. Then you have this protein structure I mentioned. You have primary, secondary, tertiary, quaternary. So this is how we differentiate between the protein. So if a primary is just is a linear, there are 21 possible combinations of the primary structure. Okay. Secondary structure is one polypeptide chain by twisting and folding created by interactions of the polypeptide bond. So that is like it's just a twisting in such a way that it will form the structure. So mainly in the secondary is alpha helix and beta pleated sheet. So still one polypeptide chain, but it twisted in a way that it becomes the either alpha or beta pleated. So alpha helix is the most common one. It is hold together by the hydrogen bond. So this is the hydrogen bond. So this is the one that holds together the formation of the alpha helix. And this is also it can be complicated, but it's still considered as secondary structure because it only has this alpha and beta formation. Okay, so that's secondary. Whereas tertiary is on a three-dimensional arrangement of protein results from interactions between R groups or between groups and the polypeptide bond. Still one single polypeptide chain, but the interactions between the R groups, and then the hydrogen bond also. So these interactions cause bending of the chain. So most enzymes and myoglobin are all in this tertiary structure. So this one. So these are the interactions in the tertiary structure. We have hydrogen bonds and then the van der Waals interaction, disulfide bond, and then the ionic bond. So all these are the one that holds together the tertiary structure. Whereas the quaternary structure, there are two or more polypeptides. So anti-primary, anti-tertiary is just one single chain, but for quaternary structure, it's two or more polypeptides held together by the same forces that hold together the tertiary structure. Okay, and then you have additional like salt bridges. These are the one that holds together the quaternary structure. And then most enzymes has quaternary structure. These are the quaternary. So this is the difference between the quaternary structure and the tertiary structure. So this is example of the hemoglobin. Like hemoglobin has 4 subunits, meaning that it's made up of 4 peptide. Okay. So it has 2 alpha chains and 2 beta chains. So this one is a quaternary structure. Whereas for tertiary structure, example is a myoglobin. So it's only made up of 1 polypeptide. Other example like your insulin also is tertiary structure. Okay. So in general, so these are the difference between all these structure. So you have the primary, this is a secondary, okay, mainly the alpha and beta. Then the tertiary protein, three-dimensional folding, and then the quaternary is made up of two or more polypeptide. Okay, so I think that one is just simple. So this is the difference between the hemoglobin and myoglobin. So structure, quaternary and tertiary, like I mentioned just now. So it's hemoglobin is made up of two alpha and two beta subunit. So hemoglobin is you find mainly in the RBC for transport of oxygen, okay, hydrogen and carbon dioxide. So oxygen towards the cells that need it, and then from the cell when it transfer the oxygen, okay, it will take up the carbon dioxide and goes back to the lung, and so that the carbon dioxide can be exhaled out from lung. Whereas myoglobin, you find it mainly at the heart and the skeletal muscle. Okay, mainly myoglobin is not for transport of oxygen. It's only to as a reservoir for the oxygen, so that when the skeletal muscle or the heart muscle really needs the oxygen, especially when you are exercising, then only the myoglobin will release the oxygen to the heart or skeletal muscle. Okay. So oxygen, because it has these 2 alpha and 2 beta subunit, each one has an ion, okay, heme. That one is the one that binds to the oxygen. So each one has one heme, so it can bind to for oxygen. Whereas myoglobin only have one heme, so it can only bind towards one oxygen. And then the affinity towards the oxygen also is different. Okay, oxygen, it has various affinity to bind to oxygen depending on the oxygen concentration. So when there's a high oxygen concentration, it has a high affinity to bind to the oxygen. But when it goes to the cells, when the cells need the oxygen, so it will release the oxygen to the cells. Whereas myoglobin, yes, it has a very high affinity. So this one it does not depend on the oxygen concentration. Later I will explain on this. Okay. And then this also it exhibits the cooperative binding with oxygen. This one does not exhibit cooperative binding with oxygen. So this one I explain. This one. So you have this oxygen
Doctor,
dissociation. Yeah.
I think is there something called Bohr shift, right? The hemoglobin has the high affinity of oxygen. Do we have to know for the Bohr shift for this chapter?
No. The Bohr effect you will learn more on the respiratory system in the systemic block.
Oh,
So here I didn't mention. I mean like you don't. It's good for you to know, but you don't need to know in detail about that.
okay.
Okay. Thank you. Okay, on the oxygen dissociation curve, so you can see here, this is myoglobin. So you have here hemoglobin. So myoglobin exhibit uh, this is sigmoidal. This is what do you call this? Hyperbolic, is it? Okay. Um, so myoglobin it has a partial oxygen of P50 is 1, so it has a very high affinity towards the oxygen. Whereas this is hemoglobin, it has a partial pressure of oxygen is 26. So myoglobin has a higher affinity compared to hemoglobin in terms of binding to the oxygen. Okay. So from here you can see that it binds straight away to the oxygen. So regardless of the oxygen concentration, it will binds to the oxygen, but only when the oxygen concentration is very low, then only, meaning that oxygen concentration, because the partial pressure is 1, so it means that when your skeletal muscle or heart muscle really depletes of oxygen, then only myoglobin will release the oxygen to the cell. So that is what it means. Whereas for hemoglobin, when there's a high concentration of oxygen, it will bind, it has high affinity. But when it goes to the cells, uh, a slight decrease in the oxygen concentration also, it already release the oxygen into the cells. So that is what it means by the this dissociation curve. And then what it means by the positive cooperative, the oxygen dissociation curve for hemoglobin is modern in shape, indicates a positive cooperative binding. So this one reflects the specific structural changes that are initiated at one heme group and transmitted to other heme groups, increasing the affinity of the oxygen. Meaning that uh, hemoglobin has a 4 subunit, right? So it will bind, when one oxygen binds to one of the subunit, it will increase the affinity of other heme to bind to oxygen. So that what it means by the positive cooperative binding. So when one is already binded to one of the heme, so it will increase the affinity of hemoglobin to bind to other oxygen until the all the heme is occupied by oxygen. Okay. Whereas for myoglobin, it didn't exhibit the cooperative binding because myoglobin only have one heme. Okay. Based on structure. Okay. Myoglobin only have one heme, so when oxygen binds to it, it didn't. I mean like it won't exhibit any cooperative binding because there's no other heme there for it to influence, and it cannot influence other myoglobin also. Okay, so those are two things. Okay, hope you understand on the why the affinity is different and why this one exhibit cooperative binding and myoglobin didn't exhibit the cooperative binding with oxygen. Okay, so those are two concepts that you need to understand. Okay, yeah.
Doctor, back on this graph, doctor, the
Mm-hmm.
graph,
This
uh,
graph.
the, the number of myoglobin and hemoglobin, is it the total number of myoglobin or is it just one myoglobin in our body? Is based on this graph.
Based on this graph, it's only I think it's only one.
Okay.
Okay. Okay. Any more question? So I hope you understand on this concept.
Doctor, I have
Okay.
a question.
Yeah.
Um, can you explain more about the cooperative binding? I don't see that.
Oh, cooperative binding. Okay. I'll explain again. Okay. So hemoglobin has a four subunit. Okay. So it has these four subunit of heme. So when one oxygen bind to the heme, it cause the affinity. What is it? It will cause the other heme to binds to more oxygen, meaning that when hemoglobin on its own, it is not very, it won't attract oxygen that much. But when one oxygen already bind to the heme, so it attract more oxygen to, or it will influence the hemoglobin to bind to more oxygen. So that's what it means by the positive cooperative binding. So when one oxygen already bind, so the affinity of hemoglobin to binds to more oxygen will increase. So it will bind to more second oxygen and then third and then the fourth one. So this is what it means by the positive cooperative binding. Is that clear?
Clear, doctor.
Whereas, okay, so whereas hemoglobin, it only has one heme, okay? So it didn't exhibit the positive cooperative binding. Okay, any more question? Question? Okay, moving on. Okay, so this is the heme that you found in the hemoglobin, or it made up of ferrum. Okay, it's bound to four nitrogen atoms. So in the center of the heme porphyrin ring, so it has this substance, methyl, vinyl, and then propanol and such chains. Okay, you don't need to remember the structure, just for you to know that heme is made up of this ferrum and is called of porphyrin ring. Okay, this is how heme is being synthesized, and no, you don't need to remember. Just that you need to remember is that it is made up of this succinyl CoA and glycine. Later, when we go to the Krebs cycle, so there's one of the substance in the Krebs cycle is the succinyl CoA. So that's one of the function of the Krebs cycle. Okay, when there's a formation of this succinyl CoA, so this succinyl CoA can be used to synthesize heme. Okay, so this is how it happens. Okay, and if there's a defect of all these enzymes, it can cause this disease, porphyrias. So in general, these are the different disease based depending on which enzyme is affected. Like if there's a deficient of this acid of this enzyme, amino levulinic acid dehydratase, so it will cause this disease. So, but in general, these disease are all these disease are called porphyrias. Okay. Mm, so mainly you need to know is that for the heme to be produced, this heme, so it needs the succinyl CoA and the glycine. And heme also is a rate limiting is a product inhibitor for this enzyme. So when there's abundance of this heme, so it will inhibit this enzyme to stop production of the heme. Okay. So these are the disorders related to heme synthesis. So in general, it's just called porphyrias. So when there's a defect, the intermediates of the pathway will accumulate. This will cause toxic effects on the nervous system. So why this porphyrias is very uh, can I say some is little, some is not, depending on the severity. It can cause a toxic effects on the nervous system and it cause neuropsychiatric symptoms such as anxiety, confusion, hallucination, and the seizures. So when these accumulated, they may be converted by light to porphyrins, which react with molecular oxygen to form oxygen radicals. So this also will cause damage to the skin. And thus, these individuals with excessive production of porphyrins are photosensitive. Okay. So these are the effects of the um, disorders of the hemosynthesis. Because when when this enzyme is deficient, it cannot be converted to heme. So one of these substance will be accumulated in our body or in our blood vessel. So these substance are mainly toxic to our body. So it can affect the nervous system. It can affect the uh, it will form oxygen radicals, free radicals. Okay, it will damage the skin. Okay. So mainly you just need to know on in general just that. Okay. Okay, what will happen if the protein structure is changed? So all of your protein, like I mentioned, it has all the mainly it is in our body is mainly the tertiary and the quaternary structure. So what happened when these structures are changed? So example that I given here is sickle cell anemia. So mainly sickle cell anemia. What happened is that the sequence of the amino acid glutamate is being replaced by valine. So this is a kind of mutation, substitution mutation. Okay, glutamate is being replaced by valine, so it causes the structure of the protein to become distorted. So it becomes a sickle cell. So what happened in sickle cell anemia is that due to this valine substitute, it causes and exposed to the hydrophobic region of the hemoglobin. So of this beta subunit of the hemoglobin. So normally it didn't have the hydrophobic region, but when this valine is being substituted, so changes will be in the beta subunit of the hemoglobin. So it become hydrophobic region is exposed. So what happens that when it becomes a hemoglobin, the hydrophobic region tends to interact with one another with its other sickle cell anemia. So they become crystallized and fiber. So the capacity to carry oxygen is greatly reduced. So when it becomes fiber like this, it cannot carry oxygen and it might cause clot to the small blood vessels. Unlike the normal one, okay, if the normal hemoglobin is just one subunit, so it can carry oxygen normally. But when it becomes like this, the formation of the sickle cell, so it will form crystallize with one another and forms a fiber, so it cannot carry the oxygen. Okay, so that's what happens in a sickle cell hemoglobin. So this is an example of if there's a changes of the protein structure. So this is one of the example. Okay, any question on this? So I hope that is clear. Okay, others, there is a protein structure. Other example is that on the protein misfolding, when the protein fails to fold correctly, so it will cause these are some of the example of the disease like cystic fibrosis, Marfan syndrome. So you can check later on this disease if you want on what happened in this disease. So I won't explain further on this disease. Anyhow, you will come across this disease later in systemic and during clinical years. And then some cancers also are formed due to this protein misfolding also. because when there's a protein misfolding, generally it cannot function normally. So that's why it can cause many disease. Other than that, some protein will form insoluble aggregates that deposit toxically. Like for example, in Alzheimer's disease, there's a deposition of the beta amyloid. So when there's formation of this beta amyloid is considered, because this is not a normal protein that is being produced by our body. So when there's this unknown protein is being produced by our body due to protein misfolding. So it can cause damage to our normal neuron cell. So which is why you have in Alzheimer's mainly it affect the cognitive. You know that the forgetfulness and all. Okay, is due to degeneration of the neuron cell. And our body also, when they detect that these proteins are different, so it considered as a foreign materials. So what happens when our body detect foreign materials? Our immune system will try to destroy the foreign materials, like the virus and bacteria that enters our body. Okay, it tries to destroy the virus and bacteria, but this one is protein. So basically, our body can't really destroy a protein. Even you create antibody, you cannot destroy the protein. So what happens is that when the immune system tries to destroy this beta amyloid or foreign materials, it will also destroy our own normal neuron cells. So which is why there's a neuronal death. So then you have all the other symptoms. Okay. Same with the Parkinson disease also. The position of this the abnormal protein is called the alpha synuclein. Okay. So that's in general what happens when there's a protein misfolding. Okay. So what caused this protein misfolding? Okay. Some that we know is genetically inherited. So for example, is Huntington, and others is sporadic. By sporadic meaning that is due to aging or the incorrect lifestyle. Like for example, the Alzheimer's, the Parkinson's. Um, some are associated with gene, but some and mostly it's not associated with gene. It's more towards the aging and it's more towards the incorrect lifestyle. Okay. Some cancer also, we can see that some cancers are familiar, genetic, but some nowadays they are mainly the sporadic, mainly due to the lifestyle and the aging. And others is due to the virus and bacteria, okay, transmissible, like the Prime disease, Creutzfeldt-Jakob disease, mad cow disease, okay. All these can cause protein misfolding. Okay. Okay, next we go to the protein classification. So we have these simple proteins, like previously on the carbohydrate and lipid also there are classification. So for protein also there are classification. So this is classification based on the simple proteins, conjugated protein, and the derived protein. Mainly simple proteins are the one that contains only amino acids, whereas the conjugated proteins are the one that has one or more non-protein prosthetic group, meaning that combines with other, like this one, glycoprotein, okay. And then this one is the derived protein. Is due to the nature of the proteins. This is not found in nature and usually found as a product of hydrolysis. So that one is on that. The other one is a classification based on the sizes, or mainly on the axial ratio, the ratio of the length to the breadth. So you have the globular protein and then a fibrous protein. So in globular protein, it is completely folded and coiled. So often globular protein, as the name implies, is often in a spherical shape. So meaning the enzymes, most enzymes are in the globular shape, and some hormones like insulin and prolactin. Then even your hemoglobin also is in the globular protein form. So some fibrous protein are in the uh, fibrous, meaning that it has a long shape. mostly is, for example, is a keratin, myosin, collagen, fibrinogen. Mainly these are the one that gives the structural role. Okay. So enzymes, I think there's no enzymes that is in this fibrous protein. So mainly the one that you found in the synovial fluid, in the skin, like in the collagen, okay, fibrinogen, and the keratin in hair. They give the strength or elasticity to a particular tissue. So there's difference between globular and fibrous. Okay, this is another difference between the fibrous protein and globular proteins. So examples here, and where it can be found. This one mainly in the cell organelles, molecule, okay, structure and function. So mainly fibrous protein is for structure, to strengthen, to impart the strength to the area. Okay. Mostly in the joints, ligaments, tendon, muscle, connective tissue, and skin, and other tissue that are exposed to a heavy mechanical stress. Whereas global proteins are mainly involved in the transporting and then antibodies. Okay, so this one can just read difference. And these are other classification according to their function. So based on the structural, contractile, transport, storage, hormone, enzyme, and protection. So there are three different kinds of classification, whether it's a simple or is a the size, the actual length to breadth, and then this one is classification based on their function. Okay, next is on the properties of the protein. Um, as usual, it has the physical properties and the chemical properties also. So this one, physical properties, has a high melting points, low vapor pressure, and have large dipole moments and high dielectric constant. So this is the, say like lipid has its specific properties like the hydrophobic and hydrophilic. This one, the protein also has this. This is the specific or the special characteristic of protein, has the have large dipole moments and have high dielectric constant, indicate the amino acids are polar. So it exhibit this zwitter ion. Later I will explain on the zwitter ion. Okay. Then these are the other properties. Isoelectric point. Proteins, they have charges, which means that they have a negative and positive charge. And they uh, in normal condition, it has a overall zero charge. Positive and the negative charge are equal, meaning that for protein or for an amino acid, it has the both positive and negative charge, but in a pH point or a normal condition, they have a overall zero charge, meaning that the negative and the positive is equal, so it cancels each other. So that is zero. But pH higher or lower than its isoelectric point, it will exhibit uh, positive or negative charge. So this also I will explain. Then solubility is soluble in water, but it's soluble in organic compounds like benzene. Things like heat. Soluble proteins have charge on the surface. It has its lowest solubility at its isoelectric point. Without net charge, protein-protein interactions and precipitation are more likely. Okay, the solubility of proteins in blood requires a pH in the range of 7.35 to 7.5. So this is based, the solubility also is based on the isoelectric bond. Next is on the protein presentation. So mainly, in specific precipitation may be used if you want to extract the protein. So addition of salt or amino sulfate is mainly used. And then changing of the pH to alter the nature of the solution. So when you add salt and change the pH, you cause the protein to precipitate. So it's much more easier for you to the protein. So mainly this is used to purify and concentrating the enzymes. Also denaturation. Uh, when it's very when the protein is exposed to a very high temperature that you cannot handle, so it will cause denaturation of the protein. So this is another specific features of the protein as it can denature. So what happens when it
Excuse me, doctor.
yeah,
I'm so sorry for cutting, but I think that you're cutting off. We can't hear you clearly.
really
Uh,
okay. I'm not sure whether it's my connection or not. But can you hear me clearly right now?
yeah, now better.
Okay.
Thank you, doctor.
Okay, so for the protein denaturation, so what happens is that when the protein is denatured, so it lost its function, mainly because when in high temperature, uh, the formation like the secondary, tertiary, and the quaternary structure that I mentioned just now. So all the binds, all the disulfide bonds, hydrogen bonds, and all, it will be disrupted by the high temperature. So this will cause the bonds to be liquefied, okay, to be changed. So that will change the shape of the protein. So when the protein shape is changed, like for example in an enzyme, so the enzyme can no longer function normally. So which is why we say that the enzyme or the hormone will lose function when it is denatured. Okay. It can be denatured either by heat, usually is by heat, or by acids and bases. When the pH is too high or too low for the protein, for the hormones or enzyme, it will also disrupt mainly the hydrogen bonds and the salt bridges. So this will also cause the changes or the formation of the enzymes and the hormones. So it can cause the protein to lose its function also. Again, same organic compounds also have metals, and then agitation also can cause disruption to the hydrogen bonds and cause the uh, formation change. Okay, this is on the chemical reactions. So these are the tests that you can do to identify the protein. Okay, um, you don't need to know in detail about this. This is just for you to know what are the tests that we can do to identify the protein. Okay, next we go to the amino acids. Is there any question before I proceed? Okay, so amino acids are the chemical units of the building blocks of the body that make up the protein. So this is the subunit of the protein. Okay, this is the definition. Won't go. Okay, the amino acid structure. Like I mentioned just now, it contains of the carboxyl group, amino group, and the different that differentiate between the 20 amino acids are the side chain. So there so the carbon, the middle carbon is called the alpha carbon. So the alpha carbon is bonded to 4 different atoms or groups of atom. It is a tetrahedral. So most alpha amino acids is a tetrahedral, but some, so we call it as chiral. So meaning that all these different groups, all are different groups. So we call it as chiral. But except for proline, have an acidic carboxyl group. This one, and glycine. Glycine has the same hydrogen group in both. Okay. So again, the peptide bond. So the different types of amino acid, um, you have a total of 20 amino acid. Then also you will learn more in the metabolism of the amino acid. Mainly the difference that I want you to know is on the whether it's glucogenic and ketogenic. Okay. And then this amino acid I mentioned, it binds together by the formation of this peptide bond. So what is the characteristic of this peptide bond? So difference with the carbohydrate and lipid, this peptide bond is the one that give the charges to the protein. So how is that? Um, Peptide bonds are quite rigid and planar. It exhibit the large dipole moment polar in the favored trans configuration. So this is what happen when there's a formation binding at the carboxyl and the amino group. So this carboxyl and the amino group is the one that exhibit the uh, negative and the positive charge. The amino group will give the amide nitrogen will give the partial positive charge, whereas the carboxyl oxygen will give the partial negative charge. Okay, so which is what we call as a small electric dipole. This will set up a small dipole moment for the protein. So this is special, the peptide bond. So this is the formation, okay, peptide bond, carboxyl group and the amino group. Amino group is usually positive. Carboxyl group is usually negative. So when we draw an amino acid or a polypeptide chain, so we usually put it in the form of this zigzag to indicate that it is in a trans configuration. Okay, so this is another example, and this peptide C and the between the carboxyl group and the amino group, it is not free to rotate. So this is another characteristic of the peptide bond. So which is why we call it as rigid. So it cannot bind freely. Okay. Okay. So any question on the peptide bond? So next is on the classification of the amino acid. Amino acids can be classified based on the charges and the polarity. Okay, so this is non-polar, polar uncharged, polar positively charged, and then polar negatively charged. You don't need to remember all those, but this one you at least need to remember, especially which one is the glycogenic and the ketogenic amino acid, and also you have to familiarize with the abbreviations, because these abbreviations is also going to use when we do the transcription and translation. Okay, this is another classification based on the essential and non-essential, like fatty acid yesterday. We also have essential fatty acid. So this one also has essential amino acids, meaning that it cannot be synthesized by the body, so you have to obtain it from your diet. Okay, so I'll go to the zither ion that I mentioned just now. Why this amino acid has the positive and the negative? Okay, uh, when there's a zither ion, meaning that at this point it has an equal number of amino group and the carboxyl group, and forms when the H from the carboxyl group in an amino acid is transferred to the amino group. So meaning that the H from the carboxyl, because carboxyl has COOH, right? So this H is transferred to the amino group NH2, so it becomes NH3. So in the normal condition of a protein or an amino acid, it will be in this form, in the zither ion form. So this one is the one that exhibits the dipole moment. So the has the positive charge and negative charge. At isoelectric point, these two charge cancels each other, so it becomes neutral. Okay. Any question on that? Hope you're clear. So at isoelectric point, so there is zero charge. So what happens when there's okay, at this zero charge, amino acid is least soluble in water, so it is insoluble and it does not migrate in the electric field. So this one I explain further what it means by migrate in electric field. So what happens when there's a difference in the what happens is there's a difference in the pH. Okay. So when there's a high pH, what is abundant? What is increased in the high pH? Which one is increased in the high pH?
An ion.
An ion. Okay. So what is you have abundant of H or reduced H?
Reduce it.
Reduced H, right? So what happened when there's a reduced H?
Alkali.
Okay. So what happened to what do you think happened to the protein?
Denature.
Before it denature? What happened? What are the changes in the
Carboxylic group like H removed, something like this.
it's more acidic? You understand by the meaning of this? The carboxyl in the zeta ion accepts a proton and the amino acid has a positive charge. But this one is lower. But if it's higher, the NH3 plus in the zeta ion loses a proton and the amino acid has a negative charge. So meaning that in a high pH, okay, this is a higher pH. So because there's a reduce in the H, so it will lose a proton to the solution, so the amino acid become a negative charge. So it becomes negative charge. But what happen in low pH when there's abundance of the H plus, excess of H plus? So the COO in the zeta ion accepts proton. So when there's abundance of H plus, so the H from the solution will bind to the carboxyl group, so it becomes positive. Okay, at the low pH solution. So that's the difference. Okay, in the normal pH or where the pH is suitable for the amino acid, so it remains zero, like zero charge. But when there's an increase of a pH or alkali, it becomes negative charge. But when there's a reduce of a pH or acidic, it becomes positive charge. Okay. So I hope everyone is clear on that. So what's the importance of this isoelectric point? So mainly what we do is that separation of the plasma proteins by charge that is typically done at a pH above of the pi of the major proteins. Thus the charge of the protein is negative. So in an electric field, the protein will move towards the positive electrode at a rate determined by the net negative charge. So this separation of plasma proteins, like in our plasma, sometimes we have to identify whether we have a lot of albumin, globulin, and all that. I'm not sure whether you've done this in your high school. Previously, we've done it to separate using the electrophoresis. This one, okay. So in electrophoresis, but nowadays it's already automated, so you can just separate the proteins. But some labs still doing this. Okay, can separate the proteins based on their molecular weight. Okay, so this setup is called the electrophoresis. So protein will be put in this well. So all these are well, this one. Okay. So protein we put here because protein is in its isoelectric point, it is zero charge. So for it to move, we need to make it negative charge. Okay. So when we make the protein negative charge, we put what? What pH we put? Is it alkaline solution or acidic solution? If we want to make it negative charge.
Alkaline solution.
Okay, so we put alkaline solution to make it negative charge. So when it's negative, as you remember, only negative will move towards the positive, right? That's physics concept, okay? So negative, when we put an electric current here, so the negative charge of the protein that has been negatively charged will move towards the positive. So the protein will be separated based on the molecular weight. By means of the molecular weight, meaning that the larger fragments will move much more slowly, whereas the smaller fragments will move much more quicker. So this is how we can separate between the proteins. So you can see the protein bands. So that's how you can differentiate between the different proteins. Because our plasma protein, it contains like globulin, what else? Albumin. So each one of it has a different molecular weight. So from there, we can differentiate. So you have this patient has high albumin or high globulin, that kind of stuff. Okay. This one, the same setting also is the same for this one is for protein. We also did this to separate DNAs. Okay, because DNA also DNA is negatively charged, so we can separate the DNA using this concept also. Migration from negative to positive charge, and then we can see the fragments of the DNA based on its molecular weight. Okay. Okay. Any question on this? So I hope you understand the concept of this isoelectric point. Okay, before I go further. Any question? Oh yeah, I forgot. Your attendance. Okay. Please sign your please fill in the form. Same link as yesterday. So the question, how many amino acids are there? How many how many amino acids that you know of? Okay, so please fill in your attendance form. Link is the same. Okay, so I proceed with the glycoproteins. So glycoproteins, like I mentioned yesterday, it has a high variable amounts of carbohydrate. In glycosaminoglycan, it has more carbohydrate than protein. But in glycoproteins, it has variable amount of carbohydrate. Some has more carbohydrate, some has less carbohydrate, but it contains both carbohydrate and protein. So they differs in terms of length. Glycoproteins are usually short, okay. Then they don't have the serial repeats. Some may be branched, some may be linear. Some may and may not negatively charged, and they have this N-link and O-link oligosaccharide chain. So again, the same. This is some of the example of the glycoproteins. This is on the O-link oligosaccharide chain. Say it's the same like previously. So this is N-link. So in addition to that, just want to focus on these two glycoproteins, okay, mucins, which we can find found at the GIT and the respiratory, and also at the reproductive tracts, and also in the membranes of the various cells. So mucins, it exhibit a high content of this glycosidic linkage, and it is negatively charged. Okay, and this mucin also, it contains a repeating amino acid sequence rich in serine, threonine, and proline. So main function of mucin is lubricate and forms a protective physical barrier on the epithelial surface and involved in the cell-cell interactions. So mainly the negatively charged is the one that functions mainly to lubricate and forms a protective physical barrier of the on the epithelial surface. Right. And the O-glycosidic linkage is the one that is involved in the cell-cell interactions. So that's mainly the function, mainly to as a protective physical barrier. And then another one is a collagen. Okay, collagen that you found in your skin is a triple helical structure, and it's also a kind of fibrous protein. It forms by the post-translational modification by addition of the galactose and galactoside through an O-glycosidic linkage. So what it means by this post-translational modification, meaning that after, if you see, remember, replication, transcription, and then translation, right? Translation is the process where the protein is being produced. So after the translation process, there is a modification, meaning that after the protein is being produced, it undergoes modification at in this galactose or galactosic glucose through an O-link glycosidic linkage. So it forms the collagen. So the most abundant one is a type 1 collagen. Okay. So this is how collagen is being formed. You know, collagen is you can find mainly in the skin. So it for the skin to become more to impart strength on the skin also. Okay, so this is how it is being formed from one precursor chain. So it forms this. That's why it is called the triple helix structure, because three collagen it binds together to form this triple helix. This is the molecule, and it forms this fibril, and then it forms the fiber. So this is the one that we found in the skin, the collagen fiber. Okay, any question? So that's generally what you need to know for the protein and amino acids, and the concepts that you need to know on the zeta ion, the isoelectric point of pH and all. Any question? Okay. If there's no question, what time is it now? Oh, 10.
10.
Do you want to start at 11 or you want to make it earlier? At 10:30.
Anything will do, doctor.
10:30, eh? Okay. So we continue with enzyme and its application at 10:30. Okay. Thank you. See you at 10:30.
Okay, thank you, doctor.
Grazie.